Yes, good afternoon, students. Join fast. So first of all, I am going to show you the video of cloning. Okay, and watch the video carefully, everyone. Yes.
If you already have an animal or plant with all the characteristics you want, then it would be ideal just to make loads and loads of identical copies of it. That is what cloning is, making genetically identical copies of organisms. Cloning animals is a lot harder than plants. You can't just chop off a bit of it, plant it in some soil and it will grow into a clone. Plants have very special areas called meristems which allow them to do that, which are full of stem cells. Animals don't have that. They grow very differently. And it took scientists a bit of time, but in 1996 a sheep called Dolly was the first mammal cloned from an adult cell. The reason, if you want to know why they were called Dolly, was actually, it's a bit of a bad joke by the scientists involved, but it was cloned from a udder cell, which is essentially a mammary gland, and a very well-known country singer, Dolly Parton, has particularly large mammary glands. So they named the sheep Dolly because it came from a mammary gland cell. Anyway, Dolly the sheep. How was Dolly cloned? Well, first a normal adult cell was taken from sheep 1, from the udder of sheep 1 that they wanted to clone. So this was a normal cell with the full number of chromosomes in it, a typical adult cell, just like me taking one of your skin cells, let's say. Okay? Now, the DNA was extracted from that cell because that DNA has the genome in it for that sheep, and that is the entire genetic code needed to clone that sheep. Now, a completely separate sheep, sheep number 2, was taken, and an egg cell was taken out of that sheep. Now, egg cells have the ability to multiply and to grow and form an embryo, so they're very, very important. But you don't want the DNA from that sheep, so that egg cell had its nucleus removed. We call it being enucleated. And the nucleus from sheep 1 was then put into the nucleus from sheep 2. Using electricity, the egg cell is then stimulated to start dividing into an embryo. And then that implanted embryo gets put into a surrogate sheep. Could be a completely different sheep, sheep 1 and sheep 2. So now we've got a third sheep involved. And after a while, the embryo grows and the sheep gives birth, and when it gives birth to a clone of sheep 1, because the only DNA involved in the whole process was taken from that initial adult cell of sheep 1. Now that process could be done in exactly the same way with a human, potentially. It hasn't been done, but there's no reason why that method wouldn't work in a human as well.
Yes, students, you can see here the first one. So how the cloning takes place. First, you can see the first, we have to choose, okay, select the adult from where the cell is to be taken. The second one, that is the egg cell, which was taken for cloning purpose. It was enucleated. Enucleated means the nucleus was removed from that. Okay, and here the nucleus which was taken, or the cell from the sheep which was taken for cloning, this actually the DNA was extracted. Only the DNA or only the nucleus was introduced in the cell. So cell is from the second sheep, the nucleus or DNA is from the first sheep. Okay, everyone have understood? And you can see the diagram also. From this you can understand the you can see the characters which are taken from the first sheep, because all the nucleus or the DNA that is carried out from this first one, only the first one. Okay, and this is now it is introduced into the cell which was first enucleated. It is only the empty cell in which the nucleus is now introduced, and now what is the next step? Here you can see, using electricity, the egg cell is stimulated to start dividing into an embryo. So now this is the further next step, starting the development by using the electricity. So it will start, the cell started dividing into, means it became multicellular part, and now the next step is that this was implanted, this cell further which was divided into so many cells was implanted into a surrogate ship. The third ship, you can see here the number 3. So this third ship is another one which is just taking part in developing the embryo. Only it is developing the embryo. इसका काम क्या है? She is not related. तो ये है biological mother, we can say, because the nucleus is taken from this one, this first number ship. Okay. Then this is only, we can say, she is landing the embryo. Okay. Surrogate ship number 3, and then ship 3 then gives birth to a clone of ship, and that was named Dolly in 1996.
Now, scientists have genetically engineered sheep so that they make medicines for humans in their milk. It's amazing. These medicines can treat things like cystic fibrosis and multiple sclerosis and potentially types of cancer. And the medicines usually come in the form of proteins in the milk, such as antibodies. But when you genetically engineer a sheep and you breed from that sheep, then although that genes you put in won't necessarily get passed on to the next generation, they might not get passed on at all, or half the next generation of males that wouldn't produce any milk anyway. But if you could clone that amazing sheep that you've made, then you can make loads and loads of copies of that sheep, which you've spent a lot of time and money engineering perfectly. Then you can mass produce some of these medicines really, really well. It's also hoped that in the future we could use the cloned animals to produce whole human organs such as kidneys, hearts and livers, which could be used for human transplants. So cloning potentially has some really, really important uses in treating diseases.
So have you watched that video, students? All of you? Okay. So this is, here you can see that this is the cloning process that takes place, and how the next part is the importance of cloning. Importance of cloning in human beings. Like we can say various parts can be cloned, and we can use it in various diseases, and we can save the people by cloning purpose. Okay. Understood now? So this was about the cloning. Now we will start with the quizy. Okay, students. हाँ. Okay, so students, you can see here. So we will continue with the quiz. Yes. So here. Yes, students, now I am copying the link. You can this to the quizy start. Okay, everyone, all the participants. Voice is low. Yes. 15 participants have joined. Come on. Yes. Do it fast. Still 16 students are left, 11 students are left. Everyone should join here. Come on. Yes. Do it fast, student. Join fast. Yes. Minimum 30 students should participate. Yes. 23 students have joined. Still, I am waiting for again 2 minutes. Where are the others? Yes, 25. Okay, 7 students are still here. Join it fast. So, what are they doing here? Yes, Shivaganga, Sonu Tiwari. Join fast. Come on. So let us start now. Sonu Tiwari have not started till now. Yes, Bhargav. we will just copy. Students, you can see here on the leaderboard. This is the leaderboard. Okay. Yes. Yes, students, you can join. Come back. So this is the result of today's quiz and the attendance link. This is your attendance link, students. Fill the attendance link. Tomorrow onwards, we will start with a new lesson. Okay. Question answers I will be sending in the group, WhatsApp group. Okay, PDF. So write it down, everyone. Copy into the notebook. Show me. Okay. Clear? Okay. Okay, students, thank you now.